Hi everyone, and I welcome you to the third episode of our series, that is Journey Through Lower Plants, Understanding Bryophytes. Okay, so in the last two videos we have discussed on the general characters and the classification of bryophytes. Today we will discuss on Riccia. Okay, so Riccia belongs to the class Hepaticopsida, and under this class we will study another bryophyte known as Markinsia as well as Bilia. Okay, so under Hepaticopsida we will be studying Riccia, Markinsia and Bilia. We will be studying everything regarding these three specimens. We will study the external morphology, the internal morphology, and then the male sex organ and the female sex organ along with its development, and then we'll study the structure of the sporophyte and the development of the sporophyte as well. Okay, so Riccia has about 130 species so far, and they are widely distributed in both tropical as well as temperate regions of the world. They are known to grow in moist and shady places, as well as on the barks of trees and on the walls as well. So they are more or less terrestrial plants, which means that they are found growing on land, except for Riccia fluitans. Okay, so Riccia fluitans is known to grow on water. Okay, it is an aquatic bryophyte. They are found floating freely on stagnant water or on slow running water. Okay, so the rest of the species are terrestrial, that is, they are found growing on land, and then Riccia fluitans is an aquatic species. Okay, and out of the 130 species found all over the world, about 33 have been reported in India. So we will start with the first topic, that is the external morphology. So we all know that the gametophyte is the main blunt body, right, in bryophytes. So the blunt body, as you can see here, the blunt body is flat and it is a dorsal ventral thallus, which means that it has two surfaces, a dorsal side and a ventral side. So the one that we can see here, that is the one that is facing towards us, the upper surface, this is the dorsal side, and the side that is facing towards the ground is known as the ventral side. So it has two surfaces. The blunt body is flat and it has two surfaces, the dorsal surface and the ventral surface, which is bostrate. Okay, bostrate means that the blunt will not grow upright. Okay, so if this is the ground, the plant will not grow upright. A plant will grow in this manner. Okay, so it is flat and prostrate. Okay, it is also ribbon-like structure. Branching of the thallus is dichotomous. So in Riccia you will find dichotomous branching. Okay, so this is the example of dichotomous branching. So dichotomous branching, it is a pattern where the main axis will divide into two equal halves. Okay, so it's the main axis will divide in two equal halves. Alright, so this is the example of dichotomous branching seen in Riccia. And also, due to repeated dichotomous branching, you'll find that the plants often take a rosette form. Okay, this is the rosette form that you can see here. This is the blunt body, but due to repeated dichotomy and also due to the presence of the plants nearby, they'll take the structure of rosette form, that is circular or more or less like a star-shaped arrangement. The thallus is usually green in color. This is due to the presence of green pigments like chloroplasts, and each branch of the thallus is provided with a thickened midrib. So on the dorsal surface you'll find the midrib present in the middle. Alright, and you'll also find the presence of a dorsal groove in the middle. in the middle. Okay, so the plant body will be somewhat like this. So in the middle you'll find the midrib, and then this furrow here, this curvature that you can find on the plant, this it will be known as the dorsal groove, or it is also known as furrow. Now on the ventral surface, this is the ventral surface. So if we flip the plant, the surface that is facing towards the ground, you will find these hair-like structures. These hair-like structures are known as rhizoids. These are rhizoids. In Riccia, you will find two types of rhizoids. These are smooth wall rhizoids and tuberculate rhizoids. And both of these rhizoids are unicellular, which means that they are single cell structure and they are unbranched. Okay, they are unicellular and these are unbranched. So there's no branching of the rhizoids. So this is the smooth wall rhizoids, and in tuberculate rhizoids you'll find bag-like projections. Okay, so these sharp points that you can see here, these bag-like projections, this type of rhizoids are known as tuberculate rhizoids. And along with the rhizoids, you'll also find the presence of scales. These scales are multicellular. They will be found on the peripheral regions, okay, on the margins of the plant body, And these rhizoids, these scales are pink in color or purple in color. This is due to the presence of a pigment known as anthocyanin. So the function of the rhizoids, it's more or less like roots, but these are not true roots because they have no vascular tissues like xylem and phloem. But they perform the functions of roots like absorption of water and minerals from the soil, and they also help the plant to anchor to the substratum. Okay, so they'll help the plant to anchor to the soil or to the bark of the trees or on the walls. So, and also this is how the rhizoids will look like under a microscope. This is a smooth wall rhizoid. And this is a tuberculate rhizoids. Okay, so these are the bag-like projections. These dot structures, these are the bag-like projections in the tuberculate rhizoids. So everything upon the external morphology. Next we will go to the internal morphology. Okay, so before that, now in order to understand or say study the internal morphology, we'll have to take a DS section of it, right? So if we take a DS section of the plant, a vertical transverse section of the plant, and then under a microscope you'll find something like this. Okay, and then this thallus will show a internal tissue differentiation, which means that the plant body is not just a mass of cells. Okay, it has specific tissues doing specific functions. All right, so on the dorsal region you will find photosynthetic cells. Okay, this upper region here, this is the dorsal region. You'll find that this will contain a lot of photosynthetic cells, and this region is known as the photosynthetic region. And then on the ventral side, on the ventral region, you will find cells that are compactly arranged without any intracellular cells. Okay, these are parenchymatous cells. You find no intracellular spaces between the cells. They are compactly arranged, and they are also known to store starch as reserve food. So this region here, this ventral region is known as the storage region. So in the internal morphology, you'll find tissue differentiation. Find that the cells are divided into specific regions. Photosynthetic region that contains pigment system, chloroplast containing cells. Along with that, on the ventral region, you will find the presence of parenchymatous cells that contain starch as a reserve food material, and this region will be known as the storage region. Alright, and then the chloroplasts are without pyrenoids. So these pyrenoids are protein. Okay, these are proteins, and then they are commonly found in algae. And the functions of these pyrenoids is for carbon fixation, and they are also known for starch, I think for starch storage, and they are commonly found in algae. So in the chloroplast of Riccia or in hepatic Copsida in general, pyrenoids are totally absent, but they are found in some bryophytes like Endoceros. Next is vegetative reproduction. Riccia is known to reproduce by two methods: vegetative reproduction as well as sexual reproduction. Okay, so we'll first discuss the vegetative reproduction. So vegetative reproduction is a type of asexual reproduction where sex organs are not involved. So the plant body itself will give rise to a new plant, okay, without any events of fertilization. No sex organs will be involved in case of vegetative reproduction. So the first point is by progressive death and decay of the older parts of the thallus. So the first point is the fragmentation that is found in Riccia. Okay, so the portion that you can see here, this is known as the apex, okay, and this region is known as the posterior end. So the plant body will grow in this direction. This is the younger part of the plant, and this is the older part of the plant. Plant will grow in this direction only. So as the plant continues to grow in this direction, the older portion will start to die and decay, and over time this portion will start to decay, and then each of these fragments will be separated from one another, and this will give rise to a new Riccia plant. This will also give rise to a new Riccia plant. So this process is known as fragmentation. The next type of vegetative reproduction is by advantageous branches. So this type of reproduction is seen in Riccia fluitans. Okay, this is a aquatic bryophyte. So what will happen is that on the ventral surface of the plant body, they will start producing advantageous branches. Okay, in each of these branches, once they are separated from the main plant body, they will give rise to a new plant. Alright, the next is by tuber formation. This tuber formation is seen in, for example, Riccia, pulpirifera, Pulpifera. This is also seen in Riccia, this color. Okay, so tuber formation is seen in Riccia pulpifera, Riccia, this color. So what happens is that after the growing season, the plant body will start producing these tubers. Okay, tubers at the apical region of the plant body, they will start producing tubers, and then these tubers will grow on the ground, and then the main plant body will start to die off, and then during favorable conditions, each of these tubers will now give rise to a new plant. And then by death of the thallus in drought condition. Okay, so these are somewhat similar, tuber formation and death of the thallus in drought condition. So what will happen is that the apical region that I've mentioned, the growing region of the plant, so if suppose this is the plant body, the apical region they will grow on the ground of that condition, that is when there is insufficient water. So the apical region will grow on the ground, and then this region will die off. And when there is sufficient water, or when the everything is favorable during ideal conditions, the apex will again give rise to a new plant. Alright. Sexual reproduction takes place by means of well-developed sex organs. The male sex organ is known as antheridium or antheridia, and the female sex organ is known as archegonia or archegonium. Most of the Riccia are homotelic. They are homotelic, meaning that they bear the female and the male reproductive structures on the same plant. So some of the examples includes Riccia, melanospora, Riccia, Castellana, and then Riccia robusta. Okay, so these are some of the examples of homotelic or monoecious forms. Most of them are monoecious forms, but some of them are heterotelic as well. And the examples includes Riccia, this color, and then Riccia frosty. Okay, so these two are the examples of heterotelic or dioecious forms of Riccia. And as the thallus matures, the reproductive structures develop on the dorsal surface of the plant in acropetal succession. So what it means is that if this is the plant body, this region here is known as the apex, okay, and then this region will be known as the posterior end. So what it means is that the mature reproductive sex organs will be present at the posterior end, and then the younger reproductive structures will be present at the younger part of the plant, that is the apex part of the plant. So as the plant matures, reproductive structures will be present at the posterior end, and the younger reproductive structures will be present at the younger part of the plant, that is in the apex region. And this is known as acropetal succession. Acropetal succession. Okay. Now let us first study the antheridium. Okay, antheridium is singular, and then antheridia is plural. Okay, so this is the male reproductive structure of Riccia. So as you can see, a mature antheridium is globular in structure. It is globular, oval, or club-shaped structure. Okay, this region here that you can see here, this oval globular structure, and the space that you can see here, this space is known as the antheridial chamber. Inside the antheridial chamber, the antheridia will be produced. Now there is an opening in the antheridial chamber, which is known as the ostiole. Okay, and then the central part of the antheridium will consist of a mass of cells, and this mass of cells are known as androcyte mother cells, and this androcyte mother cells will give rise to the male gamete, that is the sperm. And then this androcyte mother cells will be surrounded by a single layer of cells. This single layer of cells, this green shaded region, these are sterile jacket cells. Okay, they are sterile, meaning that they do not contribute in the production of sperms. Okay, and inside this you will find fertile cells. These fertile androcyte mother cells will give rise to sperms. Alright, now let's study the development of antheridium. So this male sex organ antheridium is present on the dorsal side of the plant body. So on the dorsal surface there will be a cell that is somewhat larger in size, compared to other cells, and this cell is known as the antheridial initial. This cell is also larger than other cells. It will have a dense cytoplasm. Alright, now what will happen to this antheridial cell is that it will undergo one transverse division. And after it undergoes transverse division, it will give rise to two types of cells. The upper cell will be known as the outer cell, and then the lower cell will be known as the basal cell. Okay, so two cells will be produced, outer cell and then basal cell. Now on this outer cell, two successive division will again take place, and this will give rise to four cells. Okay, and these four cells that you can see here, these are known as the primary from these four cells, these two upper cells are known as the primary antheridial cell, and then these two cells that are present on the lower side are known as the primary stalk cell. Okay, so this primary stalk cell will give rise to the stalk, and then these upper two cells, the primary antheridial cells, will give rise to the antheridium. Now what will happen is that these two cells that you can see here, these primary antheridial cells, will undergo two vertical divisions. Now vertical division will take place in this manner. Okay, so the first division will take place in this manner, and then the second division will take place at the right angle of the first division. So from the upper surface, if this is the upper surface, if the first division takes place in this manner, the second division will take place at the right angle of the first division. Okay, so if the first division is here, the second division will take place in this manner. Okay, and this will give rise to 8 cells. So this is how the cell will look like. This is how the division will look like from the above. Okay, so the first division here, and then the second division here, right? So this will give rise to 8 primary antheridial cell. Okay, 4 at the front, 1 2 3 4, and then 4 at the back. And these two lower cells, these are the primary stalk cells. This will give rise to the stalk. Now what will happen is that this primary antheridial cells will undergo a special type of division. And this division is known as periclinal division. This periclinal division is a type of cell division where new cell wall formation will take place parallel to the cell surface. So if this is the cell surface, a new wall formation will take place in this manner. Okay, that is parallel to the cell surface. Okay, so this type of division is known as periclinal division. There is another type of division known as anticlinal division. This type of division will take place perpendicular to the cell surface. So if this is the cell surface, the division will take place in this manner. Okay, perpendicular to the cell surface. So you'll find that new wall formation will take place in this manner. So you'll find cells like this, but here you'll find that division will take place in this manner, that is parallel to the cell surface. So these 8 cells will undergo a periclinal division. So periclinal division will take place in this manner. Okay, since this is the cell surface, since this is the cell surface, periclinal division will take place in this manner. Okay, in this manner. All right, See, periclinal division will take place in this manner, and then this will lead to the production of two layers of cells. The outer cells that you can see here, the outer cells, these are known as primary jacket cells, and this will contribute in the production of the jacket cells. And then the inner ones, these are known as the primary antheridium cells. Now these primary jacket cells will undergo anticlinal division. So anticlinal division, let us just draw one of the cell. Anticlinal division, since this is the cell surface, anticlinal division will take place perpendicular to the cell surface, right? So new wall formation will take place in this direction only. Okay, so all the primary jacket cells will undergo anticlinal division in this manner, and then the primary antheridium cells that you can see here, this will divide in all planes. Okay, they will divide in this direction, they will divide in this direction as well, and finally this will give rise to these primary jacket cells. After the anticlinal division, they will give rise to the jacket cells. These are single layer jacket cells, and these are also sterile. Okay, sterile cells, they do not contribute in the production of sperm. And then these cubicle cells that you can see here, after the division of the primary antheridium cells, these cubicle cells, these are known as the androcyte mother cells. So this is androcyte, this is an androcyte cell. These are cubicle in structure, and this will undergo one final division. Okay, the division will be in a diagonal form, and then each of these cell will metamorphosize and give rise to an antherozoid. Okay, antherozoid, or we can also call it as sperm cell. This is the male gamete, that is the antherozoid, and each of the antherozoids will have two flagella, 1 and 2. Alright, so the sperm in bryophytes are always flagellated, and they will always be biflagellated. Okay, biflagella meaning two flagella. So that's how the antheridium is developed from a single cell in the dorsal surface of the plant body. Alright, the female sex organ is known as archegonium. The archegonium is a flask-shaped structure. It looks somewhat like a flask that is connected to the archegonial chamber with the help of a short multicellular stalk. Okay, it's not present here, but there will be a presence of a short multicellular stalk here. And this archegonium is differentiated into two regions. This lower region is this solan region is known as the vender, and then this upper elongated region is known as the neck. Okay, the neck is composed of 6 vertical rows of cells. So from the top view, it will look somewhat like this: 1, 2, 3, 4, 5, 6. Okay, this neck is composed of 6 vertical rows of cells, and each of the rows will consist of 6 to 9 cells. Okay, so this region, this is from the side view, and then from the top view it will look like this. Okay, it will consist of 6 vertical rows of cells, and each of the rows will consist of 6 to 9 cells. And the tip of the neck is known as cover cells. So these are the cover cells. So there will be 4 cover cells. As we can see from the side view, there are 2 at the front and then 2 at the back. So this is this structure of a mature archegonium. Now we will study how this archegonium is developed. So just like antheridium, archegonium is also developed from a single cell, a single superficial cell, a cell that is larger in size when compared to other cells. And these cells, since it is larger, it will protrude out on the dorsal surface, and this cell, just like antheridium, is known as archegonial initial. So this cell is larger compared to other cells. Now what will happen is that this archegonial initial will undergo one transverse division, and this will give rise to two cells. The lower cell is known as the basal cell, and the upper cell is known as the outer cell. Now this outer cell is also known as the archegonial mother cell. Okay, so this single cell will give rise to the entire archegonium. Okay, it will give rise to this entire archegonium just from one single cell alone. So now this outer cell will undergo 3 vertical eccentric intersecting division. Okay, so let me write it down here. Okay, so from the side view it will look somewhat like this, but from the top view it will undergo 3 divisions. Okay, 3 vertical divisions in this manner, and that will give rise to 3 peripheral cells. These cells, this region here, this will be known as the peripheral cells, and then in the middle you will find the presence of a primary axial cell. This is the primary axial cell from the side view, and then from the top view this is the primary axial cell. Now these peripheral cells will undergo anticlinal division, okay, in this manner, and this will give rise to 6 cells, and these cells are known as the jagged initials, and each of these cells will give rise to the 6 vertical rows that we have discussed earlier. Okay, and so each of these cells will undergo a transverse division, and then in this manner, and this will give rise to the next cells that you can see here. All right, now what will happen? So at the same time, when these cells are dividing, this primary axial cell that is in the middle will also start to divide. Okay, so first division will be a transverse division, and this will give rise to 2 cells. The first cell, this primary axial cell, will undergo one transverse division. Okay, and this will give rise to upper primary cover cell, and then the lower one will be known as the central cell. Okay, now this primary cover cell will undergo 2 vertical divisions. Okay, so the first division will occur in this manner, will divide in this manner from the side view, and then from the top view it will go on one vertical division, and then the second division is also a vertical division that is right angle to the first. Okay, and this will give rise to 4 cover cells that you can see here. So 2 at the front and 2 at the back. Okay, so the cover cells will look something like this. Okay, so cover cells are already formed. Now this central cell again will undergo one more transverse division, and this will give rise to 2 types of cells: the vendor initial cell and the primary vendor cell. Okay, this vendor cell will keep on dividing transversely. It will keep on dividing in this manner, and this will give rise to the neck canal cells. Okay, these neck canal cells will be formed through these vendor initial cells. Alright, and this primary vendor will undergo a division, and this will give rise to the egg that you can see here. This will give rise to the egg and a vendor canal cells. Okay, so this is how the archegonium is developed from a single archegonial initial. So what will happen is that this archegonial initial will undergo a transverse division and give rise to outer cell, basal cell. This outer cell will undergo 3 vertical eccentric intersecting division, and you will form a structure like this that will consist of 3 peripheral cells and then 1 primary axial cell. These peripheral cells will divide and give rise to the 6 vertical rows of cells, that is the neck region, and then this primary axial cell will undergo a transverse division and give rise to primary cover cell and central cell. This primary cover cell will give rise to the cover cells, and then these central cells will keep on dividing, and this will give rise to the egg, that is the female gamete, as well as the neck canal cells. Okay. Now during fertilization, what will happen is that this neck canal cells along with the ventral canal cells, they will start to disintegrate. Okay, in the absence of in the presence of water, these neck canal cells and the ventral canal cells will disintegrate, and you will form a mucilaginous substance, okay, somewhat sticky or slimy in nature, and they will start to absorb water. And once they start to absorb water here in this region will start to expand. Okay, and due to the force of this cover cell that you can see here, the outer part will be separated from one another, and this cover cells, once it is separated, they will open up, and then the male gamete, that is the sperm, will enter and then fertilize the egg. Okay, and then zygote will be formed, which is diploid in nature. All right, and this zygote will give rise to the sporophyte. Now since zygote is formed, this zygote will grow in size, okay, and it will occupy the entire space, and it will soon start division. Okay, so the first division is a transverse division. In bryophytes it is usually a transverse division, and it will give rise to 2 cells, cell number 1 and 2. Both of these cells are equal in size, and after this it will undergo another division in this manner, and this will give rise to 4 cells, 1, 2, 3, and then 4. And the third division will be a vertical division. Okay, since I cannot draw here, the division will occur somewhat like this. Okay, from the top view in this direction, and this will give rise to 8 cells. Okay, and once the 8 cell stage is formed, which is also known as the octant stage, they will start to divide periclinally. Okay, so periclinal division will occur in this manner. All right, and this will give rise to 2 layers of cells, one outer cell and then the other is inner cell. Right, the outer cell is known as the epithecium, the one that you can see it here, and the inner cells, those mass of cells, are known as the endothecium. Okay, now this epithecium will undergo anticlinal division in this manner, anticlinal division, and this will give rise to the sterile jacket cells. Okay, this will give rise to a single layer jacket cells, and then this endothecium that is present inside, they will divide in all planes. Okay, they will divide vertically, transversely, in all directions, and it will give rise to sporogenous cells. Okay, these sporogenous cells will undergo meiosis, and this will give rise to spores. Okay, the spores that you can see here, the spores are covered by a single layered capsule wall, and then the outer covering is known as the calyptra. Each of these spores, they will germinate and give rise to the gametophyte. The gametophyte plant body. And this gametophyte plant body, they will produce sex organs on the dorsal part of the plant. The male sex organ, that is antheridium, and the female sex organ, that is archegonium. And then they will produce the male and the female gametes. They will undergo fertilization, and then zygote will be formed. In the zygote, again they will start to divide and then give rise to the spores, and then this spores on germination will give rise to gametophyte. Okay, so this is the structure of a Riccia sporophyte. In most of the sporophyte of bryophytes, you will find that there will be presence of three regions known as the capsule, seta, and then foot. Okay, but in case of Riccia, the sporophyte is represented only by capsule. Seta and foot are completely absent. Okay, so this is the structure of the sporophyte. It is globular in structure. It has a single layered capsule wall, and then it is enclosed by a structure known as calyptra. Okay, calyptra. Alright, and foot and seta are completely absent in case of sporophyte of Riccia. Now these are the two books that I've used: Studies in Botany, and then a textbook of Botany by Singh, Pandey, and then Jain. So if you're interested, then you can go to the library and then use this book. Okay, you can prepare your notes from this textbook. I feel like this textbook should be sufficient to create a good note. And if you're interested in purchasing this book, then I've mentioned the books in the description. Okay, so that's it for Riccia. We have covered the external features, internal features, the sex organs, and its development as well, right? So if you have enjoyed the video, then I request you to kindly like it and share, and then please do subscribe to my channel as well. Okay, and then in the next video we'll discuss on Markensia. So just like today, we will study a complete detail. Okay, we'll cover the entire detailed structure of Markensia and its reproductive organs as well. Okay, so see you in the next video. Bye.